Okay, we're back. Part B, Chapter 12, Holes Anatomy, Part 4 on vision. So this is looking now at the retinal cells, the retinal neurons, and there's lots of different cells involved in the retina. We mostly just think of cones and rods, but there's other things that play a role. There's ganglia that are supportive. Notice in this picture, the weird thing is here is light is entering and striking these cells where the rods and cones are here at the back of the eye. And this, again, is the choroid coat providing nutrients for these rods and cones. But in between are all these accessory cells whose names are not important, but they help to modify impulses. So they deaden certain information, so you can block certain signals. You're not constantly flooded by information. They inhibit, for example, or amplify certain pieces of information. And then you send that information back this direction through the optic nerve. So the names of the cells are not important. It's a little bit more complicated than I like, but it's showing you the retina. Now the layers of the eye, just a refresher for you. The outer tunic is the sclera and also the cornea. And then the middle tunic involves the choroid coat and then also the ciliary body and the iris. And also not mentioned here is the lens. I don't know why, also part of the middle layer. And then the inner tunic is the retina, which is for photoreception and transmission and so on. Okay, so when light enters, it refracts. It doesn't reflect like you would see in a mirror. It refracts or it bends, like the effect of looking into an aquarium and the fish looks bigger or smaller because the light bends as it enters the water. And it depends on the density of the medium. Different densities, things will bend more or less. Okay. So in each different region of the eye, light bends more or less depending on what part of the eye it's going through. Now we can adjust how light bends by adding lenses. Okay, in general, the physics of it, light converges when it passes through a convex surface. So like this, light that enters at all these different angles, all these different spots, will bend in towards the middle and converge at a certain spot. Okay, for us, this is how our eye works. Okay, and with our cornea and so on, and we want this convergence to occur on the fovea centralis. Here we have the opposite. We have a concave surface, and as light enters, it causes the light to bend out. You do have some concavity involved with the eye because of the lens is a biconvex surface. So it's convex here and convex here. And as a result, light is going to bend in and then out depending appropriately and as it works to converge. Convex lenses cause light to converge. Concave, they diverge. And then when the light from an image is passing in through all these structures, it then gets processed or placed on the retina in the back of the eye, and all this information gets flipped upside down. And then as it's sent to your brain flips it right side up. So in reality or theoretically, everything you're seeing is upside down, and then you flip it right side up. Not exactly, but it's flipped, and then you flip it back again. Okay. The image focus on the retina is upside down and reversed from left to right as well, kind of like you see in a mirror. So you have to adjust that with your eyes. So my question to you would be, what if you had a contraption that could flip your vision, let's say, upside down, so that everything you saw was upside down, and you wore that for a year, never took it off. After a year, when you take it off, would everything now in the world be upside down for you? Or would you still see things right side up? What would be the effect? And you can imagine the same effect flipping left to right if you had a contraption. And we do have those types of things, of course. These are glasses that alter your perception. So the two main visual receptors are rods and cones. Rods are long and thin. They're bipolar. And they have light-sensitive pigments, specifically called rhodopsin, and hence the term rods. They're very, very sensitive to light, rhodopsin is, and thus rods. So they provide vision in dim light. When you can barely kind of make things out, there's not a lot of light available, your rods are dominating. And you can see shapes and patterns and so on, but you can't see color. It's colorless vision, and you see largely outlines. Cones, on the other hand, are short and blunt. They contain light pigments called erythrolabe, chlorolabe, and cyanolabe, and these produce vision in bright light and thus sharp images, and also in color, of course. Those are your cones. Here you see the rods. Many rods are tied to one single sensory nerve, whereas each cone is tied to its own sensory nerve. So cones give you far more specific vision, whereas rods is more of a blunt kind of less specific, less focused or detailed image. Rhodopsin, as we mentioned, is light sensitive. Now, when you come out of a movie theater and you step out into the light, you just see this white blankness. You can't make out anything. And this is partially because your rods, which had dominated previously with rhodopsin, when they're exposed to light, the rhodopsin denatures, and it doesn't work anymore. So temporarily, the rods are not functioning and your cones have not activated here yet. And thus you temporarily can't see. So rhodopsin is very, very light sensitive. Cones, on the other hand, they're all different pigments, okay, are affected by different wavelengths of light. And the color you perceive, it depends on which cones are stimulated. Again, erythrolabe is for red. Erythro means red. Chloro is green, so chlorolabe is the pigment that responds to green light. And cyano is blue. Cyanolabe responds to blue light. Here you see rod cells. They're bipolar cells passing information. Here's the nucleus, just to show you an example. Stereoscopic vision. If you look like this, it's very difficult to see depth perception. Okay, you can identify things and their depth by their size in relation to other objects, but if they're by themselves, with one eye, you cannot see depth. And as they move, you can't see depth unless they change size. Two eyes provides two different viewpoints, view angles, slightly different images that are processed on the retina, and as a result, you combine that information in your brain to give you stereoscopic or 3D vision. And we'll investigate some of this on Thursday as well. This is the visual nerve pathway. Each eye sends information back through the optic chiasm and then through the thalamus and into the occipital lobe in the back of your brain. There is some crossover from each eye, but not completely. Okay, about half of them. So we all know that as you get older, your eyes go bad, your ears go bad, and so on. So specifically going back to hearing, when you get old, your hair cells in the organ of Corti begin to get damaged over time due to it causes hearing loss, and then the nerve pathways to the brain start to become diminished. They degenerate, and then tinnitus. Tinnitus is that hearing sound that you hear occasionally, and this is tied to that hearing loss, of course. What happens when you get old with your eyes? Yes, it sucks to get old. Your eyes start to dry out. You get floaters. Sometimes you start to see these little things going across that are not actually on the front of your eye. They blur your vision only in certain spots, and these are crystals that have formed in the vitreous humor. And so they block the light coming in, and you get these patches of blurriness or incomplete vision. The lens slowly loses elasticity, and you have an inability to focus. You might also get glaucoma or cataracts. Glaucoma is a increase of pressure behind the eye or within the eye, and as a result, you start to feel pressure and pain, and you get headaches and things like that. Cataracts is a loss of the transparency of the lens, and as a result, the world starts to turn gradually more and more yellow. You can have surgery to replace your lenses. They put in a new lens that still can accommodate, and the world becomes clear all of a sudden again. Pretty amazing surgery, in fact. I've had family members have surgery for cataracts. And then macular degeneration, the macula lutea and the receptors and so on slowly start to degenerate in the back of the eye. What happens as you start to have messed up vision, or you're born with messed up vision? Okay, so here we have refraction disorders. First of all, take a look on the left side here. Look in the middle. This is a normal eye. Light enters, it bends and converges right on the fovea centralis. Here's the point of focus, perfectly on the retina. If your eye is too long, it's a little oblong. We call this myopia. And the point of focus here is not actually on the retina. It's inside or before the retina. And as a result, the light that hits the retina has already diverged back out. And so everything is a little bit blurry. The opposite is also the case where you have hyperopia. Your eyeball is too small. Okay, and so the light is converging behind your eye, and thus everything looks a little blurry. So depending on if you are myopic or hyperopic, you would use one of two different lenses. Okay, here you have a concave lens where it diverges the light to make it where previously the light was. Here's the uncorrected point of focus. You now need to make the light diverge even more to focus deeper or further back on the eye because your eye is longer, so you would use this concave lens. And if your eye is too small, you want the light to converge earlier, so you would add a little bit more convergence by using a convex lens so it converges sooner onto the back of the eye, onto the retina. Those are how you would correct two different vision problems. And on that note, we are finito. I will see you later.